Now we move on to mutation. So from this lecture, we move on to the session 3 and 1, which we're describing mutation and population structure. So what we're going to learn today, we are having a basic introduction and evolution of the term called mutation and how this mutation term varying definition over the time. And we see sort of differences between the mutation and the term variant. And variant frequency, which represent the population structure, and we'll see some examples on that. And lastly, we'll have several genomic studies understand population structure. So first topic is about the mutation. So mutation, or we call variant, is a change of nuclear sequence in an organism. And as I mentioned several times in previous classes, a mutation is basically an allele in the context of modern genomics. Or in other words, we can say that genetic variant is an allele in the context of modern genomics. Mutation, as mentioned just before, is also called genetic variant. And also mutation is called polymorphism, or mutation is also called genetic variation. I can say that the genetic variant or genetic variation is quite common alias for mutation, but in some contexts we also use polymorphism. It really depends on when the papers or research document has been constructed. And also more recent context, we use the genetic variant or genetic variation. This is more like a synonymous. People sometimes use genetic variant, sometimes use genetic variation. Genetic variant is more general format, but people trying to have, different, varieties. That case, people use the genetic variation. And change, as I mentioned here, change it can be interpreted in the context of population. So basically, when you describe change, it's basically you compare something with others, right? So in this case, you compare your nucleotide sequence to others' nucleotide sequence. Then we can say that here is some change. And lastly, population can be defined by mutation, and also this population and mutation, this relationship has long been studied in the genomics and human genetics. So first thing we can think about is the introduction of the term commutation. In 19th century, there have been debates on how evolution works. So we briefly talked about this before. So the time people trying to describe why we are different and sort of differences in the context of the social structure. But also in the meantime, people are very interested in natural science. So basically why different biological organisms, they different to each other. So when we describe such pattern, we collectively say evolution. And when we describe evolution, people has a different perspective. So some people thought that mutation can take place suddenly. So in other words, we can say that sortating. Or some people believe that the evolution is sort of gradual process, right? So basically, the people we call sortationists, they claim evolution takes suddenly and change instant transition into the new species. So it's something, sudden appearance. But, in contrast to them, gradualists like a Charles Darwin, they believe that the process of evolution takes slow and it can, we can think about this situation given we see large, variance of traits in a population. Because, gradualist perspective is that, if the variant, if the evolution takes suddenly, then we only see few combinations of phenotypes. But, as we can see, many different traits and their variability is actually has very wide distribution rather than few, data points, right? So that's basically how gradualists think about the, evolution. And one of the paper in the time was Hugo de Vries. He is definitely the sortationist. He observed the new species of evening primrose, and that one of the flower he made observation has different phenotype, and that different phenotype certainly arose in his experiments. So that's basically in line with the circadian. So that was the 1901, and Hugo de Vries gave that new phenotype, new form of flowers in his experiment. He called that the mutation, right? So mutation is sort of the Greek word to make a permanent change. So that's basically the description. So the time when we saw the new forms of flowers, and he thought that that's probably sort of the new species, and that situation has to have description in some way, then he called mutation. And this term was later evolved with the study of gene. So in early 1930, the concept of gene had become more concrete because of Thomas Morgan, right? And genes were regarded as indivisible units of inheritance. So people thought that this gene on a certain chromosome might explaining sort of these, evolution and this mutation situation, right? So they thought that the genes can be defined in terms of few aspects. So first one is hereditary transmission, genetic recombination, mutation, and gene functions, right? Then later, people still, supporting the Charles Darwin's idea, and collectively we call them Darwinian, right? And those Darwinian, they thought that mutations do something on the traits, but they believe that the natural selection is sort of the primary force of evolution. So whatever mutations still can have impact on phenotype, but still, given the large variance of the traits in a population, probably the natural selection, swept out those variants in certain population. In the meantime, there are people supporting Darwin's idea, some people supporting the Mendel's idea, and we call them post-Mendelian geneticists. They still believe that the mutation do something, great than natural selection, right? And some people like William Baisden, Hugo de Vries, and Thomas Morgan, and those people believe that one mutation, one gene can do something in evolution. And one of the very interesting idea aligned with this sortationist and post-Mendelian was Richard Goldschmidt. So Richard Goldschmidt is known for hypothesis called hopeful monster. So what he described, he basically proposing some models about the macroevolution throughout the macromutation. So macroevolution means that the new species we never seen before appeared, and those appearance is by macromutation, right? So what is the macromutation he defined in his theory was developmental macromutation. So mutation in developmentally important genes could produce large phenotypic effect, right? So something like a gene can impact on some wing formation in a certain insect, and that wing formation makes a new branch or totally new shape of that will lead to the new species. So that case, when the organism developed in certain situation, that gene is responsible for the wing development, and if the mutation happened within that gene, then that organisms can have a new phenotype and ultimately has the new species, right? In that case, Goldschmidt thought that this macromutation can impact on the macroevolution, and that will lead to appearance of new species, and that's sort of the situation of we call sortation. So in other words, we can say that they change early development and thereby cause large effects in an adult phenotype. And is that really true? We can, think about any, supporting, research on that. And there's one example. So here is one gene called FOXP2. So this is the gene responsible for chromosome regulation, transcription regulation, and this gene looks like a fork head, literally fork head, and it bonds to DNA, and they do something in a situation DNA replication or DNA transcription, right? And this gene is surprisingly well conserved across the vertebrates. And the marmoset has this protein. And before the marmoset, like a mouse, also has this protein. And rhesus monkey, they have that, chimps has it, and obviously I have and you have it, right? And this protein is very conserved. That means that FOXP2 protein, amino acid consequence is quite similar to each other. But, when the species has a new branch, which means the different speciation, and some position of these amino acids are different. So one example is when you look at the human FOXP2 and when you look at the chimps FOXP2, we have the different amino acid in certain sequence. Let's say T303N, which means that the protein 303 position, it changed from T to N, right? But 3 to 5 is the amino acid change from N to S. So if the mutation happened at this position, and if the mutation changed to amino acid in this two position, that will lead to the different functions between chimps and human. And what is this FOXP2 known for? Is this FOXP2 is involved in language development, right? So how people, came across this hypothesis. So in many years ago, when people research people with language delay, so some people didn't have, full development of language in certain age, and they collect those people and compare with, normal language development people, and they found that several mutations found in FOXP2, and they thought that FOXP2 might be working in a language function. And after they study FOXP2 over and over years, and they, found out that some of the amino acid has changed between these species, and between the species, they're not just changing this position. If you compare with the marmoset to chimps and monks, and they have the different changes on these 652 positions. And these things impact on each species language. In other words, we can say vocalization for animals, right? So this protein is involved in a vocalization, and that will ultimately impact on the language development. This is just examples of primates, but if you see like a birds, you can also see different, amino acid change and mutation in FOXP2 as well. So, I mentioned that this FOXP2 is sort of the very interesting hypothesis for now. It still needs some several, years of research to make a, solid idea about, FOXP2 is really impact on language. But what I can tell you is that this is sort of the examples of we can think about the macromutation in human evolution. And likewise, we have another interesting evidence on macromutation, which is the gene duplication, right? So once you look at the gene, we have so many genes in our genome, and these genes are not, randomly distributed. So what that means is some genes are having similar functions because they originate from the similar ancestry gene, like we, in a human population. So if you look at the one gene, let's say some genes located in here, and if you have new mutation when you have a gamete cells, it can lead to make a new gene by duplication, right? So as I mentioned before, some of the mutation can happen as a just one nucleotide or two nucleotides or several nucleotides, but sometimes somehow some nucleotides they just duplicate certain position to here. So this is like I just drew the very short box, but this is actually like a 10 kilobase, right? And this long chunk, they just make a new duplication like in here, right? So this can also happen in when we make a gamete cells. And most of the situation, we can say that this is non-functional because, this, new mutation is mostly neutral. So we cannot think about new function. But sometimes some of these sequence, very conserved and very important, and this sequence is just, copy and paste in a new position, then we can say that the conserved gene, they make a new thing. So can make a, two, conserved genes, or we can just say conservation. Or sometimes when you make a new, new sequences by duplication, it can make a new function. So we say that neo-functionalization, right? So this gene is responsible for, let's say, metabolic situation. And once these things is duplicated in a new position, this gene can, work as a, hormonal pathway gene because the constitution around these genes can have the different regulatory elements or different, transcription factors. So it can have a new functions than previous, the sequence. Or sometimes, some of very partial of these original sequence can be functional. So in that case, we can say that it's a subfunctionalization. Or sometimes when it makes a new duplication, it can also have very specific function. So we can say there's specialization. What I want to say is when we have a reproduction, which means that making new gametes, there is always a chance to make a new sequences by mutation, right? So that case, we can introduce a new function. And one of very well-known examples is the development of synapse. So here is a synapse one we can expect from our brain. And this synapse is basically originate from certain cell functions, right? So when you look at the eukaryotes doesn't really have the synapse, but they have some cellular compartment looks like a synapse. So what is synapse? So synapse is like a two brain cells, they, connect to each other and they sending signals to each other, right? But in a very old format, this is like a primate, like a vertebrate on here, right? But in an evolution time, in a very mundane time, in this time we have eukaryotes, and those organisms, they don't have the synapse, they don't have the complex system. They only have very simple unicellular organisms, but they communicate through the certain chemicals, right? So this is the cell 1, and here is the cell 2, right? So they sending some chemicals to make some signals, right? So they forming the signaling formate network. And this relationship is like a very initial version of synapse in a eukaryotes version. But once they have more complex systems by this gene duplication, they have more and more systems, and ultimately they're forming very, intricate synaptic systems in a brain cells. So that's basically done by the gene duplication, macromutation, has happened throughout the evolution time. So this is also we can think about like an hopeful monsters Goldschmidt proposed in early days. Okay, so there was a bit of examples. Then we move on to other, you know, um, you know, mm, advanced in mutation term. So later people talking about the Darwin's idea, and those people we call neo-Darwinism, and like Ronald Fisher, Charlotte Wright, J.D. Haldane, Theodore Dobzhansky, and those people, still supporting the Darwin's idea in 1940. So they're basically saying that natural selection plays important role in, evolution, then mutation, and, sometimes we can create a new characteristics by genetic recombination, but these people still believe that natural selection creating the variation in of the trait in a population and ultimately impact on evolutions. There's some parts quite plausible because Ronald Fisher's or Shore Wright, those people are not technically biometricians, but they really supporting idea about like a mathematical formations and always describing the distribution. So they probably supporting Darwin's idea. And all of these, debates between Darwinians and Mendelians, after 1940 to up to, even these days, they still having, conflict views. But these things too complicated. But one thing I really want to empathize is Mutugimura's idea on a neutral mutation. So literally, Mutugimura, in 1970, what he proposed at the time was neutral mutation. So in other words, means that most mutation we see in a human are neutral, right? So that's basically his idea, right? So why he claimed this idea in 1970? Because when he calculate the rate of the new mutation, we can expect it from biological organisms, and that is quite high than his expectation. And he found that every generation they having, you know, a number of mutation, right? And this, given that this number of mutation, if the mutation has to something like a beneficial or harmful, then the trait variance we see in a population has to be affected severely by those, mutations, right? But as you can see that the populations you know, in our society is quite limited and seems like, people are quite resilient over the generations. So they're not really disappeared. They're not swept out by, you know, reproduction. They're always maintaining certain numbers. So he thought that the mutations are actually very neutral. So that's really important things, right? Why this is important. So when I, teach mutation to people before the genetics class, maybe from general biology or your high school genetics, when you learned about the mutation or when it comes to mutation, you might feel that mutation is either harmful or beneficial, right? You probably see some examples like when the mutation happen, then the organisms are not you know advantages on certain environmental changes, and they ultimately subject to natural selection. Or you might see some examples like a bacteria, if the mutation appeared and bacteria has become mutant and they make a resistant in certain, situation and they made a positive selection. That's sort of the examples you learned from the textbook, but it's still valid, but it's very exceptional, especially when it comes to human genetics. It's very, very, very exceptional. It's very exceptional. So what I meant is, when we see the mutation in our genome, as I mentioned several times in previous classes, we have 3 million mutations, right? So given these numbers, the 3M mutation has to do something according to textbook biology. Then we expect that we all have very, very unique and very, very different appearance to each other. But it was not, right? we have the same two pairs of one pairs of arms, one pairs of legs, right? And one pairs of ears, and we only have one brain, something, something, right? So that means that our mutations segregating in modern humans, we are actually very similar to each other. That means that these large number of mutations do something, but not drastically introduce a new phenotype as we see in bacteria, right? So that's the thing. You have to change your perspective, right? And also when we learned about the polygenic model, we talked about the different genetic components like a additive factors, dominant factors, right? When you really want to understand the additive factors, that means that very little contribution to a single trait, right? So 3 million allele can impact on one trait that has to happen throughout the very little contribution, very little contribution of an allele. So that is in line with the idea that the mutations are actually neutral, right? So that's the thing you have to keep in mind. Okay, so then we also have varying definition of mutation variant polymorphism. Mutation, as I mentioned, this is an old term, 100 years ago, and people initially described the situation for permanent change in the evolution process. Do you agree with that? I don't. Because, the examples we see in the Hugo de Vries, we see the new phenotype appearance. In that case, we can say that this is a permanent change, but the mutation we have in an every generation is just so large number, and those mutations do not do anything, right? They very like a neutral. They can do some additive impact, but they're not really driving new phenotypic change, right? So we kind of really say that this is sort of change, right? Because it will gradually impact on something, but it's not like a, status transition between two things, right? So mutation has sort of, sourced from the Greek word implicate, permanent change, but this might not be true, right? So in more recent years, people has to describe this, mutation with more general idea. So people, introduce a new term, which is the variant, right? Or specifically, we call genetic variant, right? So what is the genetic variant? This is the describing a change in the nucleus sequence, right? And as I mentioned, the nucleus sequence may not permanent change. So that's why I more support genetic variants. And this variant often called genetic variant, most likely, and genetic variation also likely, and variation as well. And another term people use is a polymorphism. This is an old term as well, and describing a variant with frequency above 1%. And broadly, people use this term describing if you know some frequency in a certain population, then we say polymorphism. Okay, so here is one example how we represent the frequency of variant across the different population. So as we see last time in a dbSNP database, you see this is the dbSNP ID. You can see the different frequency of variants in this population. So if you look at the European, it's around 23%. And if you look at African, you see 66% or something like that. And why we see the different frequency between population, because in a modern human, we have explained by out of Africa theory. So what this means that in an old times, probably 200,000 years ago, specifically we have like 170,000 years ago time. So that time, most people sitting around this African continent were very tiny population. they move out to Europe, they move out to Southern Hemisphere, they move out to Asia, and they cross the Bering and they move to America. That's how, modern human has their history. And within this time, definitely, African versus non-African people here. And they have the different compositions of the variant, and that is still reflected by frequency, right? So in other words, we can say that the non-African population have different variant frequency because of different bottleneck and long migration history, right? So you probably remember the question I mentioned last time in an Hardy-Weinberg equilibrium was that how the allele frequency was determined in a very initial population, right? Because dominance not really impacting on the allele frequency, right? So dominance not really correlate with the allele frequency. So the answer now I can give you is that when you do a migration, right? So here has like a, 1 million people. We don't know exact numbers. And very tiny number of people, they move to other places. And those tiny numbers, like in here, they made up the allele frequency in that population, right? That's the important things, right? So some people, they migrate to Arabia Peninsula, and some people move to Europe, and some people move to Asia. And those, people move to new place, they holding the allele frequency and they move out to other places. So in other words, if you see the allele frequency, you can have the data that genetic pool has much larger size in the African population. And if you look at the non-African population, you have the small genetic diversity because of this, history. So that's the thing when we're describing how the allele frequency determined at the first time. This is sort of the answer. Okay, so variant frequency or allele frequency, the same meaning, can characterize the population structure or genetic diversity. And some factors like migration, bottlenecks, they interact with our traits, and they're making trait variation in population. And also the factor is represented in variant frequency, right? So one example is like if you see some allele frequency of hemoglobin gene, and that allele frequency is responsible for taking more oxygen in blood, let's say that. And if you look at those allele, you will see that the high allele frequency in Tibet population compared to other Asian population. Why that happen? Because some people, they migrate to Tibet in early days, and some of the people has the allele responsible for, high oxygen um, high oxygen capture, and they can survive in the Tibet environment because it has a high altitude, right? But if you see others like doesn't have those don't have these allele, and they probably selected out by the high altitude situations, right? So after many generations, you see high allele frequency of those blood oxygen things in the population, and that will make a representation of population structure in the ancestry, right? So this is like a relation between the natural selection and the allele frequency, right? It's more like it's not like the situation when the Tibet people settle down that area and they have a new mutation, cope with this high altitude. It was not, right? More like how those people segregating in that position and they interact with natural selection that is the represented in allele frequency. And also, we have very important terminology you have to understand as when we describe an allele, you can describe in these two. right? So first one is the wild type allele, or we can say the common allele. Or we can say that variant allele or alternative allele, right? So this is very, very, very important terminology. It's probably more important than dominant allele versus recessive allele. This is the description of the modern genetics, and this is the description we used in the genetic data we use in these days, right? So let's look at the wild type allele first. So definition is that there is a single prevailing allele usually present more than half of the individuals in the population. And wild type allele comes from common ancestor or founding population. This is no doubt, this is so true. This is by definition is true, but in the data, sometimes there's an exception. I can give you an example. This is the thing you have to understand. So when you look at the database, like DBSNP, you probably see that the wild type allele frequency is like a 20%, let's say wild type. And if you look at the variant allele, it's the 80%. It doesn't really make sense to this situation, right? Why these things happen. So at the very first time when they make a database, they measure, let's say they measure the 10 people, right? And they measured 10 people and they see how many people has an wild type allele and variant allele. Let's say this is an A and this is a C. And they found that the A allele appeared as a 8 people and C allele was a 2. So at the time when they're creating the database, they see that the A allele, which is a wild type allele, is the 80% and variant allele is the 20%, and they create the database. But once people study more and more, and let's say in 2020, they collected 40 million people and they measured this allele frequency. And these 40 million people, they found that A allele is the 20% and variant allele is the 80%. So it's an opposite. When they measure that with much larger numbers, more recent data set. So it turns out to be false, right? So you might think that we can simply update these numbers in a database, but it's not easy because we have so many, entries so far and database not really changing their, numbers and their record easily. And also the database collects so many different sources of the data. So they want to keep the original tracks when they collect at the very first time, right? So they decide to keep the original record, but they just updated it, right? So when they call the white tablet at the very first time, they still use that numbers even for now, right? But what I can say, this is sort of the exception, but these are just exception. Not many things happen like this. Mostly when we measure the variant alternatively, we just see sort of expected pattern. So what is the variant allele or alternative allele? This is literally, this is different from wild type allele and change in the nucleotide sequence of DNA. And we can specifically call variant, right? And development of genomic technologies, we see more and more variant and more alternative allele, and we have more opportunity to study population structure. So, okay, so I will give you these things first. So this is sort of the evolution of genomic technology. Then we see very early time, we only see 1 or 2 people, sorry, around 10 people at a very early time. But now we have like millions of people genome in the world. And you will see more and more examples afterwards. Okay, so when you're describing these allele information, we use the correlate theory to track down how alleles is segregating to modern humans, right? So when you see very early days, some people with certain allele and how these things is inherit to next generation and segregating to the current generation. Sorry, this is sort of the opposite. So this is the common ancestor, right? And they have some allele and they segregating to current population. So let's assume that this is the European and this is the like a Northern Europe and this is like a Southern Europe and Northern Europe like a Finnish versus like a Norwegian people, something like that. And these things how we can describe the allele in a different time. And when we're describing this covalent theory, we say that this coalescence process happen as a stochastic process. This is very important scientific jargon. So stochastics means that the random process, right? So our expectation when where these alleles segregate to next generation, we thought that this process happen as a randomly, right? So why we assume that this process happen randomly, right? Because this random process, which means the stochastic process, is a null situation. Do you remember the null? Null hypothesis is that the natural population has to happen in this way, right? Why we assume this way? Because in reality, in the real world, the things not happen as this as we assume, right? So when you describe something, looks like deny the random process, like a non-random process, we thought that there must be something. So we set up the hypothesis, we set up the null hypothesis, and then we measure some experiments and see those observations reject the null hypothesis or not. Then we can prove that some non-random thing, non-stochastic things happen in certain population and an allele, right? So our basic assumption is that the things like an allele in a population happen randomly and segregate randomly in this process, but some of the situation was not, right? So this is the way we approach to population structure. Okay, so, and this is an examples of when we see the genetic mutation in our genome. So, as I mentioned, that our genome is like a 3 billion base pairs, and this is just very, very tiny chunks of our DNA. And if you see the DNA, there's a red things like a genetic variant. It has to happen like an A, but it changed to G, or it has to happen as a C, but it changed to A. So when you study modern genomics, you see like this and you mark up this. It's not, right? It's just kidding. Um, the computer do these things for you, right? If human wants to do these things every day, you're gonna be nuts, right? So computer, do this job for you, and they're trying to find some genetic variants compared to other reference, and they're trying to find the genetic variant, right? Not by you, but this is, how data looks like. Okay, so people trying to find the population structure using genomic information. So one of the very early efforts was HAMMET project. So HAMMET project is we call, oh, did I put the full name? This is the haplotype mapping project. So we shortly call HAPMAP. So HAP is haplotype and MAP is literally map. So what they want to do is they want to see the genetic differences across the different ancestry, different population. So very early days, they look at the haplotype rather than allele, because haplotype is it is the abstraction of an allele, right? The representation of an allele. We're going to learn in later sections. So they look at those abstraction at the first time, then they measure actual variants afterwards. That is the 1,000 Genome Project, right? Because they see clear population structure throughout the HAMMET project, and they decided, okay, so let's look at much larger numbers, like 1,000 people. So they decide move on to 1,000 Genome Project. Then throughout this project, people finally got to know that variant frequency uniquely represented in each population, and we can find the population structure. And genomic data is very useful research to find the genes associated with disease and genetic variant in patients, other than the population structure. And from these lessons, many different countries, they decide to generate the national level public health purpose database, which is the National Genome Project. They have different names, by the way. So this is one example. They represent the population structure using the allele frequency, and they see the here African population, right? So AFR is then African, and they see the American population on here, like in Indians, and they see East Asian here, and they see Europeans here. And they compare the AA cohort, which means that the African ancestry, and they align those samples into this plot. And the African people, they align with the African population, right? So with with the genome information, you can easily infer the genetic ancestry from each people, right? And this is the same example, but it's for the Europeans, right? Okay, so one thing you have to remember is that when I describe the population in the genetics class, it's mostly describing the ancestry, right? So ancestry is the thing which is an population identify genetic data. But we have the different synonyms like a race or ethnicity. This is the term describing participant response or their self-declaration, right? So some guy, they participate, 1,000 genome project, and then the person, he declared himself, I'm a European, white man. But when we analyze his data, he turns out to be you know Latin American background, right? And they probably has some European background, but very heterogeneous in his DNA, right? So when we're using the DNA information, we are more in-depth, observations, how the population ancestry background mixed in his DNA rather than self-declaration, right? These things is important in two ways. First one is I put here in Korean. First thing is in our Korean language, race, ethnicity, ancestry, and population has only one word. That's very embarrassing, but we only has one word. So whenever I wrote genetics topic subject in Korean, I felt very upset about that because these four words are a little bit different in different contexts. But so that's for Korean students, I recorded these things in Korean language again for these things. And second thing is why we need to look at the ancestry using the genomic information. This is like a racism, or this is like a discrimination based on the DNA information. Some people might feel that, and we really has to be careful about this direction. But one part, very important things and very useful thing is that when you know about the genetic ancestry, you use that information to find some genes or alleles responsible for certain traits or disease, right? So let's look at that diabetes or like a LDL cholesterol. And those things, let's say, is very specific to East Asian population. And if you want to find out the genes underlying these cholesterol traits, then you have to use the information about the genetic ancestry. You have to either compare Korean to Korean, or you can compare the ancestry information, then you select out the very similar ancestry background to compare the case versus control for this disease study. So that context, we use the genetic ancestry to make up the proper experimental design and trying to implement those information in drug development as well. We'll deal with these topics in a genetic association testing in later sections, just for the a brief introduction about why we use the genetic ancestry in this context. It's not for the discrimination, it's not for the racism, it's more for the we want to make a biological interpretation of certain traits in the context of ancestry. And also, in other words, ancestry really affecting on allele frequency. And if you want to use the allele frequency for the disease study or trait study, you always consider ancestry background. Otherwise, you won't see anything, right? So this is the thing we have to keep in mind. Okay, and a few projects we can think of is first one is the Decode project. This is from Iceland in 1996 by Kari Stefansson. And the Iceland is an interesting place because they kept all the familiar history since they migrate to Icelandic territory. And using this genealogy, the family information, they can develop some genetic studies in very early on. So in 2002, they first proposed the recombination map of human genome. So, think about the recombination map. It was introduced in Drosophila 1930. But in human Iceland people, they use these many layers of pedigree. They can find out the recombination map in early on, right? And also they found out the de novo variant in very early days. So this is like a textbook change findings, and Icelandic people, they contribute these things by their genetic database. And a few more examples I mentioned many times. UK people was brilliant, and they, generate so many genomic data in early on from 2006, and they, integrate their data generation with the medical care, and they track down all different healthcare information and resident genomic information, and they're trying to answer what is the cause of disease, what is the genetic association of certain trait, blah, blah, blah. And that was the huge, collection of research over many times. And another, the notable national genome project is the FinGen. Literally, it's from the Finland. And Finland is located in Europe, but it's a little bit different from mainland Europe because they have different histories of isolation and different religious or social background. And those things is represented in their allele frequency and their disease prevalence. And all of these things has to be understood. So they decide to make a national genome project and they generate some data. And that was also very important research for the research. And Korean government, we just studied the National Genome Project. We about 20 years later than UK or Iceland, even way later than Finland, but brilliantly, we now move on to the project. So National Biobig Data Project, we call this name. It starts from the 2004 April, and it will collect the data over 9 years. And hopefully we can use this data set to understand our different genetic background in our Korean society. And commercials, we can think of this, allele frequency and population structure. And one example is an ancestry. If you look at the United States residents, you can use their genomic information and they track down where they're from in terms of the genetic ancestry. Okay, so a few quick summary. So mutation, as I mentioned many times, is the change in the nucleotide sequence, and various terms like a genetic variant, polymorphism describe mutation, and their frequency reflects the neutral selection. And many different large-scale genomic projects is going on to understand population structure. And we move on to the actual the concepts and details of mutation, and hope you see in the class.